Hello everyone, and welcome to Biopedia. Today, we're going to be discussing two-component systems, which are molecular systems used by bacteria as well as some archaea and eukaryotes to detect stresses and respond to them. Two-component systems consist of two separate proteins, which are known as a sensor kinase and a response regulator respectively. The sensor kinase sits within the membrane, with a portion facing into the cytosol and a segment facing into the periplasm of gram-negative bacteria, or the extracellular milieu of gram-positive bacteria. We'll doubtless come back to the differences between gram-positive and gram-negative bacteria in the future, but for now the only relevant difference is what the external section of the sensor kinase faces into, as outlined above. In brief, gram-negative bacteria have both an inner and an outer membrane, as opposed to just one plasma membrane, and the periplasm refers to the space between these two membranes. Anyway, this sensor kinase can sense a specific stimulus, whatever that stimulus might be. If it is detected, the sensor kinase phosphorylates itself, or autophosphorylates, at a specific histidine residue. For this reason, the sensor kinase is sometimes also known as the histidine kinase. However, I'm going to be sticking to sensor kinase throughout the rest of this episode for the sake of clarity. Once this phosphorylation takes place, the phosphate group is then transferred to the second protein, known as the response regulator. This is a cytoplasmic protein. Phosphorylating the response regulator activates it. Now, the reason why this is relevant to the bacterium is that response regulators are usually DNA binding proteins. Its binding acts to either positively or negatively regulate certain genes, and thereby allow the bacterium to respond appropriately to the stimuli present in its environment. It should be noted that, although the majority of response regulators bind to DNA, others control enzyme activity by acting after translation, or interact with other proteins within the cell. Although there are lots of case studies, we're going to look through one as an example. Specifically, we're going to be discussing the osmolarity regulation of the gram-negative bacterium Escherichia coli. As an aside here, osmolarity refers to the concentration of solute in a solution, in this case surrounding the E. coli cell. E. coli has two different pores within its outer membrane, called OMP-C and OMP-F, which allow metabolites to get into the cell. OMP-C is, importantly for our purposes, a smaller pore than OMP-F, as will become relevant in a moment. The sensor kinase in this example is called ENV-Z, and the response regulator is called OMP-R. Once high osmolarity is detected, ENV-Z autophosphorylates, and the phosphate group is transferred to the response regulator OMP-R, which is in turn phosphorylated at a particular aspartic acid residue and activated. OMP-R is a DNA binding protein, and its activity acts to repress the transcription of the gene encoding OMP-F, and promotes transcription of the gene encoding OMP-C. Thus, at relatively higher osmotic pressure, more OMP-C is synthesized. By contrast, more OMP-F is made at lower osmotic pressures. Because net water movement is always from an area of low solute concentration to an area of high solute concentration in order to even out the relative concentration, having smaller pores would prevent the cell from losing too much water. However, this system only works properly as a way of adapting to stimuli if it can be reset. To that end, phosphatase activity acts on response regulators in two-component systems, removing the phosphate and thus deactivating response regulator proteins at a constant rate. In fact, this dephosphorylation is often undertaken by the response regulator itself, rather than a separate protein. Now, this dephosphorylation usually takes place at a slower rate than the sensor kinase phosphorylates the response regulator. However, if the sensor kinase activity drops and phosphate addition to the response regulator stops, phosphatase activity eventually removes the phosphates and resets the response regulator back to its original unphosphorylated state, essentially acting to reset the system. So, that's the basics of two-component systems. Thank you all for listening. Feel free to get in touch at the show's email address for any questions, comments, or topic suggestions for future episodes. Until next time, have a great week, everyone.